Multiple sclerosis could be caused by bacteria, and scientists might have just identified two of them. In a rare study of 81 identical twins, where only one sibling had MS, researchers identified two gut bacteria, Eisenbergiella tyyi and Clostridium, as likely triggers of the disease. Since identical twins share the same genes, the researchers could isolate environmental differences, focusing on gut microbes from the small intestine's ileum, where immune activity is high. These two bacterial species consistently appeared in higher numbers in MS-affected twins, and were confirmed again in a larger international study with over 1,000 participants. To test whether the bacteria were actually causing MS, scientists transplanted gut microbes from the twins into germ-free mice. The results were striking. Over 60% of mice that received bacteria from MS-affected twins developed spinal lesions and signs of paralysis, while fewer than 10% of the control mice did. In several cases, spikes in Eisenbergiella tyyi or Clostridium growth directly preceded the onset of symptoms, especially in female mice, matching MS's higher prevalence in women. Both bacteria belong to a larger family usually seen as harmless, but this study suggests that under certain conditions, like low dietary fiber, they may shift from helpful to harmful. These bacteria appear to feed on mucus sugars in the gut lining, potentially weakening the barrier and triggering inflammation. Eisenbergiella tyyi also produces compounds like ethanol and succinate, which are known to stimulate immune responses that can damage nerves.
Genetically engineered bacteria can self-destruct inside tumors, releasing chemicals that can ultimately kill certain cancer cells. In a new study, scientists created a special version of Salmonella bacteria that does exactly this in colon cancer tumors, and the results in mice were promising. The researchers used a weakened strain of Salmonella typhimurium that naturally travels to tumors, then engineered it to self-destruct once it builds up inside the cancer. When the bacteria break apart, they release a protein called light, which sends strong signals to the immune system. This signal kicks off a chain reaction that activates important immune cells and builds tiny immune centers called mature tertiary lymphoid structures right next to the tumor. These structures help the body's immune system recognize and attack cancer cells more effectively. The treatment boosted cancer-fighting T cells and increased the number of helpful immune cells in the gut, reversing a harmful shift in cell types usually seen in colon cancer. Compared to using regular Salmonella, the engineered version caused much stronger immune responses and led to slower tumor growth, longer survival, and in some cases, full tumor control in mice. This approach works by using the light protein to activate a specific immune pathway, but when the researchers tried it in mice that were missing either the light